Hello, welcome to Cerebral Lectures and welcome to the second part of the glycolysis video. We have discussed the reactions of glycolysis pathway in our last video. In this video, we will discuss the regulated steps and the hormonal control. Stay with me. High energy states always inhibit the process, whereas low energy states always activate the process. But why? Glycolysis is all about producing energy like ATP from glucose. So when there are already so much ATP in the body, we will definitely not need more ATP. So there will be a stop to the glycolysis. But when the body is in a state of low energy, there are very less ATP available for the body to carry out its normal metabolism. Our body will require more glucose to push it into the process of glycolysis to produce energy. So low energy states activate, high energy states inhibit. This will help you in memorizing the regulated steps. Here is a summary of the regulated steps. This is the first one, this is the second one, and this is the third one. We are first of all going to look at the first regulated step carried out by the hexokinase or the glucokinase enzyme. They are actually the same thing, just different isomers. So glucose is converted into glucose 6-phosphate, and this is the first regulated step. I have added an illustration here to make it easy to understand. Glucose is converted into glucose 6-phosphate and then glucose 6-phosphate into fructose 6-phosphate. Some books state fructose 6-phosphate regulates the enzyme, some say glucose 6-phosphate regulates the enzyme. So they are the same thing, no need to get confused. Here the illustration says fructose 6-phosphate, so we will go by it. Otherwise glucose 6-phosphate is also okay. So there is a regulatory protein for glucokinase enzyme. The protein is named glucokinase regulatory protein. This protein binds to glucokinase enzyme and keeps it inside the nucleus of the cell and obviously inactivates it. So when there are like very high levels of fructose 6-phosphate, these levels will activate the glucokinase regulatory protein. This protein will bind to glucokinase enzyme and inactivate it and keep it inside the nucleus. When the glucose levels again begin to rise in the cell, this is a signal for the glucokinase regulatory protein to leave glucokinase enzyme. And then this enzyme comes into action and converts glucose into glucose 6-phosphate and pushes up the entire pathway of glycolysis. This is pretty simple. I hope I've made it easy to understand. The glucokinase regulatory protein binds to glucokinase enzyme and inactivates it. The process of inactivation is activated by fructose 6-phosphate, whereas glucose 6-phosphate inactivates the process of inactivation. There's so much inactivate. Anyhow, it is so simple. Back to our regulated steps list. We have discussed the first one. Now we have the second regulated step of glycolysis carried out by the enzyme phosphofructokinase 1. This is the reaction where fructose 6-phosphate is converted into fructose 1,6-bisphosphate in the presence of phosphofructokinase enzyme. Regulation is done in two ways. First of all is the regulation by energy levels, and secondly we have the regulation like covalent regulation. As I mentioned earlier, low energy signals will activate, like AMP is a low energy signal, it will activate the reaction, whereas ATP and citrate are considered as high energy signals. They are going to inhibit the reaction. Easy? Now the second one is the covalent regulation, which is done through this thing, fructose 2,6-bisphosphate. Let's look at a pretty illustration to understand this process. This one here is a figure explaining how the substance fructose 2,6-bisphosphate activates glycolysis. Let's imagine that we have glucagon. We are imagining right now. So glucagon will activate the adenylyl cyclase enzyme. Cyclic AMP will form, which will activate the protein kinase A, and it will phosphorylate this bifunctional phosphofructokinase 2 enzyme. When it will be phosphorylated, this enzyme, when it will be phosphorylated, it will go into an inactive state. But right now in our body, we don't have glucagon. We have insulin, because insulin is high after a meal and insulin has to activate glycolysis. We have insulin high concentration. Obviously the whole pathway of this protein kinase A and its phosphorylation will not be occurring anywhere in the body. So there will be dephosphorylation. Instead of phosphorylation of the bifunctional enzyme, there will be dephosphorylation of this domain, phosphofructokinase 2 domain of this bifunctional enzyme. This domain, phosphofructokinase 2, will become active and it will convert some of the fructose 6-phosphate into fructose 2,6-bisphosphate, then goes and activates glycolysis. I hope I explained it very well, how insulin kicks up the production of fructose 2,6-bisphosphate, which then activates the process of glycolysis. We have discussed two of our regulated steps, and now it's the turn for our third regulated step, which involves the enzyme pyruvate kinase and also is the final step of glycolysis. Pyruvate kinase is also regulated in two ways. Firstly, by feed-forward regulation, and secondly, by covalent regulation. Here, look at the activators of the pyruvate kinase enzyme. First, we have AMP, a low energy signal. As stated earlier, it will activate pyruvate kinase, and the other thing is fructose 1,6-bisphosphate. The process by which an intermediate of the pathway activates another enzyme within the same pathway is known as feed-forward regulation. Fructose 1,6-bisphosphate, which is the intermediate here, activates an enzyme here, pyruvate kinase. This is the feed-forward regulation of pyruvate kinase enzyme. Some high energy signals like ATP, acetyl-CoA, they will inhibit the enzyme pyruvate kinase, whereas AMP, fructose 1,6-bisphosphate, will activate the enzyme. I have explained the feed-forward regulation of this enzyme. Now let's have a look at the covalent regulation, which is done in the presence of glucagon hormone. Let's imagine that glucagon is high. Glucagon is a fasting hormone which is present in the states of fasting when energy levels are low, when a person is not taking his meal. So this glucagon will activate adenylyl cyclase enzyme. ATP will get converted into cyclic AMP, which will then activate protein kinase A. The main function of protein kinase A is to make some phosphorylations. It will phosphorylate an active pyruvate kinase enzyme into an inactive pyruvate kinase enzyme, ATP. ATP will donate a phosphate group to pyruvate kinase and it will become inactive. So now the conversion of phosphoenol pyruvate into pyruvate will not occur. This is how high levels of glucagon regulate the enzyme pyruvate kinase. A very simple regulation. These two methods have been discussed and I have tried my best to make this regulation easy for you. But before we jump to the other part of the video, let's have a look at the summary of these regulations. Here, I have made some mind maps as the summary of the regulation of glucokinase. First step, I told you, Glucokinase is the enzyme of that step. It is regulated by glucokinase regulatory protein. I illustrated it very well in that figure. Second step is carried out by enzyme phosphofructokinase. It has two ways for its regulations. First of all, intracellular energy levels. Secondly, through fructose 2,6-bisphosphate. I explained both of these pathways. Third one is the pyruvate kinase enzyme, which is the final enzyme of the pathway which converts phosphoenol pyruvate into pyruvate. It has two ways for its regulation: feed-forward regulation and covalent regulation. Again, I explained these pathways really well. Now let's have a look at the hormonal regulation of glycolysis. First of all, insulin is released after a meal, and its main aim is to decrease blood glucose levels and deliver more ATP and more energy to the body. Glucagon is the main fasting hormone which increases blood glucose levels throughout the body. These are the three regulated steps of glycolysis. Insulin will activate each of the regulated steps, whereas glucagon will inactivate each of the regulated steps. This is really simple, really plain, and I have done my best to explain this to you, and I would be really jealous if I ever come to realize that someone has explained these regulations better than me. So if you have any suggestions, leave them in the comment section. I will definitely have a look at those. Take care. Bye.